This presentation will be on Monensin and its modes of actions in supplemented cattle diets by John Gentry. First off, what is Monensin? Monensin is an antimicrobial. It's in the form of a carboxylic polyether antibiotic. This antibiotic is isolated from Streptomyces cynomonas. In the picture to the right, you can see a slide culture of Streptomyces species. This is not the specific one which produces Monensin, but it gives a general understanding of what they would look like under a slide. It was first approved by the Food and Drug Administration in the 1970s. It was originally used for control of coccidia in poultry, but they figured out that it could be supplemented in ruminant diets and had a purpose there. So now it is commercially sold as Rumensin and has been since 1976. So to fully grasp Monensin and what it does, we first need to understand what it is. And it's an ionophore, which are feed additives used in ruminant diets to improve feed efficiency and body weight gain. They're antimicrobials, which means they go in and disrupt the microbial population that exists within the rumen. The way they do this is they act to disrupt the ion concentration gradients across the cell membranes of the bacterial cells, specifically the calcium, potassium, hydrogen, and sodium concentration gradients. And the picture to the right is just a little illustration about how they might disrupt the ion concentrations, but this is a mammalian cell. How do ionophores work? Ionophores are highly lipophilic, which means they have effects at the membrane level. We know that membranes consist of a lipid bilayer, which means they are easily able to pass through since they are lipophilic. It acts as a shield when transporting ions across the cell membrane. This acts to delocalize the charge, which allows it to pass through. And the organisms that it impacts, they have to spend extra energy in an attempt to control their own disrupted ion concentrations. This use of energy is ultimately what leads to their death. And to the picture over here, we have a little representation of how the ionophore surrounds the ion and allows it to travel through the membrane. What bacteria do ionophores impact? Monensin has the greatest impact on gram-positive bacteria. You can see them in the picture to the right on the top. These are ones that have a single peptidoglycan layer that is porous. It allows Monensin to quickly reach the cell membrane and pass through, carrying these ions and disrupting their gradient. Gram-negative bacteria, on the other hand, are pictured on the bottom right, and they are separated from the environment by a lipopolysaccharide layer, an outer membrane, and a periplasmic space, as well as a peptidoglycan layer. Monensin is bound by both gram-positive and gram-negative bacteria, but the effects are only shown to occur in the gram-positive bacteria. The purpose of Monensin supplementation is to improve feed utilization, reduce or prevent acidosis, and lower methane production. Monensin has several modes of action. A mode of action is how Monensin goes about achieving its purpose. Four of these that I'll talk about are the change in gas production, change in VFA production, change in ammonia production, and the change in ruminal retention time. Change in gas production. Methane is an environmental concern. It's been brought about in new legislation such as the Green New Deal. Methanogenesis is coupled with acetate production. It makes it more efficient, as we can see in the picture to the right from Dr. Calloway's lecture. We get more ATP from acetate producing bacteria because methanogens provide a reducing sink for hydrogen in acetate production. Methanogenesis results in about a 12% loss of feed energy. This is a loss of feed efficiency as well, because methane is a wasteful product of fermentation. Monensin can be used to decrease this wasteful methane production, and it's been shown that it reduces the production by about 27 to 30% in feedlot steers. You would think that methanogens would be directly inhibited, but they're not. Monensin actually acts on the bacteria that are responsible for cross-feeding the hydrogen to the methanogens used to produce methane. So the next way that monensin acts is a change in the VFA production. Propionate is a gluconeogenic VFA. It's more efficiently used by the animal than either acetate or butyrate, which was demonstrated by Hungate. Propionate is also the most reduced VFA, so it has the most amount of hydrogen on it compared to the others. As I mentioned before, acetate production is coupled with methanogenesis, and many acetate-producing bacteria are sensitive to monensin, which means they can't produce acetate. Treatment of monensin increases propionate. Propionate is the most reduced VFA. The rumen needs a place to dump hydrogens that are not being utilized by methanogens to produce methane, so therefore we have an increase in propionate production. Acetate production is inhibited, as mentioned before. Less acetate means less methanogenesis. As propionate is increasing and acetate is decreasing, the A to P ratio decreases, leading to a more efficient gain. The next mode of action is a change in the ammonia production. Ammonia is a compound that results from fermentation of peptides and amino acids derived from protein degradation. Obligate amino acid fermenting bacteria produce ammonia rapidly, and ruminal fermentation of amino acid yields an excess of ammonia. To the right, we can see a picture of what all goes into ammonia production and bacterial proteins. So ammonia is produced in excess, which means it's a wasteful process. Anything that is wasteful leads to a decrease in feed efficiency. Ammonia is in excess because it can't even be used by bacteria fast enough to go to producing their own proteins or producing DNA. So degradation of protein to produce wasteful ammonia represents an inefficiency in overall protein utilization. Monensin impacts this by inhibiting the growth of OAA bacteria. This reduces ruminal ammonia production by as much as 50%, which leads to less waste. The final mode of action I'll talk about in this presentation is a change in the ruminal retention time. We know that the rumen is a consortium of bacteria, protozoa, and fungi. Well, monensin acts to reduce overall feed intake. When feed intake declines, the dilution rate decreases. This results in a longer period of time that the bacteria and the protozoa and fungi are able to sit there and act on the feedstuff. The fermentation rate and the extent of digestion increase in response to increasing surface area. So the more time that the animal spends ruminating, it increases the extent of digestion and leads to a greater feed efficiency. In the chart to the right, we can see how the amount of feed digested in the rumen is impacted by the retention time. As I said before, monensin is commercially sold as rumensin, and they advertise that this product, based on the studies that they've done, increases overall feed efficiency by 4% and provides a net return of $23.14 per head. I'll summarize the information in this PowerPoint. Ruminal fermentation is impacted by 4 key modes of action when a diet is supplemented with monensin. Number 1, the change in gas production. Number 2, the change in VFA production. Number 3, the change in ammonia production. Number 4, the change in ruminal retention time. Each of these have been studied exclusively, yet it's understood that it is the net effect of all of these which come together to warrant monensin supplementation. Overall, this product provides a greater feed efficiency and reduces environmental waste. One thing I did not stress enough in this presentation is how monensin is used to decrease acidosis in cattle and bloat. That is actually how I came across this topic when discussing with Dr. Calloway about how to reduce show heifer bloat. He recommended monensin, and that spurred my interest to go on and research this project. I found it very easy to research, and there's a plethora of information and studies that I could have talked about, but I tried to summarize what I found to be the most important and interesting.